Welcome to my presentation on Human Immunodeficiency Virus, otherwise known as HIV. HIV is a disease that originated in chimpanzees. It was spread to humans in the 1920s from a confrontation between a chimpanzee and a human in what is now the Democratic Republic of Congo. At that time, people in that area often hunted and ate chimpanzees. This hunting and the injuries derived are likely the cause of the spread of the disease through blood. It likely began as the Simian Immunodeficiency Virus, known as SIV, was spread and mutated into the Human Immunodeficiency Virus that we know today as HIV. You may have noticed that SIV involves simians, which include monkeys and apes, not chimpanzees. Before chimpanzees ever had SIV, monkeys had it. Chimpanzees, however, are predators of monkeys. It has been determined that chimpanzees preyed on monkeys carrying two different strains of SIV, the red-capped mangabeys and the greater spot-nosed monkeys. Within the chimpanzee, these two strains mutated, creating a third strain called SIV-CPZ, which very closely resembles HIV in humans. This strain has the ability to infect other chimps as well as spread to humans. There are two different types of HIV, HIV-1 and HIV-2. HIV-1 is the one you probably have heard of that came from chimpanzees, is very infectious, and responsible for most HIV-related deaths. HIV-2, on the other hand, was spread to humans from sooty mangabey monkeys, is far less infectious, and progresses more slowly. Because of this, it infects far fewer people and is only found in a few countries in West Africa, including Mali, Mauritania, Nigeria, and Sierra Leone. Within HIV-1, there are four different strains, M, N, O, and P. Each has a slightly different genetic makeup. This aligns with the hunter theory, as each time that the virus was passed between chimpanzee and human, it developed and mutated slightly differently, resulting in multiple different strains. HIV-1 group M is the strain today that is most studied, as it is the one that is most prevalent and is responsible for most HIV infections today. The first known case of HIV was in 1959. It was verified with a blood sample from a man living in Kinshasa, which is part of the Democratic Republic of Congo. There are other previous death reports that lead researchers to believe they were HIV-related but have not been verified. Using that first verified sample, scientists constructed a family tree of HIV, allowing them to determine that the original zoonosis occurred around 1920. Kinshasa now has the most genetic diversity in terms of HIV strains, which reflects the number of times SIV was spread from chimpanzees to humans. Kinshasa and the area surrounding is filled with numerous transport links, including roads, railways, and rivers. There was also a growing sex trade that existed in this area around the time that HIV began to spread. The combination of the high population of migrants, along with the sex trade, catalyzed the spread of HIV. The first cases of HIV were reported in the U.S. in 1981. There were 5 cases reported from previously healthy homosexual men. Within a year, there were 270 cases of severe immunodeficiency among gay men reported, 121 of whom died. These first cases involving homosexual men served to create a shame and stigma associated with the disease that has taken many years to reverse. In 1944, HIV was the leading cause of death for all Americans between 25 and 44 years of age. The disease spread globally and is declared to be a global epidemic by the World Health Organization. To date, more than 32 million people worldwide have died from HIV. HIV can be transmitted from person to person through certain bodily fluids from an infected person, including blood, semen, preseminal fluids, rectal fluids, vaginal fluids, and breast milk. These fluids result in transmission if they come into contact with the mucous membrane, damaged tissue, or are injected into the bloodstream. HIV is commonly spread through vaginal or anal sex with someone who has HIV, sharing drug injection equipment such as needles with someone who has HIV, as well as from mother to child during pregnancy, labor and delivery, and through breastfeeding. HIV is a single-stranded RNA virus, making it prone to mutation. This helps explain the numerous strains that exist of HIV. After exposure to infected fluids, HIV can enter the body. HIV enters a host cell through membrane fusion. The membrane of HIV acts like a vesicle and fuses with the host membrane so the virus can gain entry inside the cell, and then the protein capsid breaks open, releasing genetic material. When HIV enters the cell, it is viral RNA. An enzyme called reverse transcriptase then converts that viral RNA into viral DNA. Transcription will then occur using the cell's own enzymes in the nucleus. Then, the HIV mRNA is transported to the cytoplasm, where it is translated into long chains of viral proteins and enzymes. HIV is positive sense, meaning that the RNA can be directly translated into protein. As HIV continues to replicate, it spreads all over the body, which is when people begin to get sick. The symptoms of HIV can slightly differ between males and females. Not everyone displays symptoms initially. If they do experience symptoms, they include fever, fatigue, headaches, sore throat, muscle aches and pains, joint pain, and swollen lymph glands. These symptoms can also be accompanied by HIV rash, which appears as concentrated pink and red bumps that form large patches, normally on the upper half of the body. These symptoms are the body's response to HIV spreading from the point of infection to the body's lymph system, which triggers an inflammatory response. After a period of time, the immune system is able to take control of the virus. The chronic stage then ensues, which involves the virus slowly dismantling the body's immune system over months and years. Antiretroviral therapy, otherwise known as ART, is used to treat HIV. You may be wondering what retroviral means. Well, HIV is a retrovirus, meaning that instead of transcribing its genetic code from DNA to RNA like a normal virus, it does this backwards, thus transcribing from RNA to DNA. Antiretroviral therapy accounts for this and is designed to work against it. ART is used to suppress the HIV in the body, aiming to bring it down to detectable levels. Scientists have studied the mechanisms by which HIV replicates, and ART is used to specifically target certain stages within the virus's life cycle. There are 7 different classes of antiretrovirals in existence today that interrupt at the stages of attachment, fusion, reverse transcription, integration, transcription, assembly, and budding and maturation. Within these classes, there are 39 different antiretroviral drugs available. These drugs do not eliminate the virus, they inhibit its ability to replicate. By proceeding with treatments like this, your immune system is not additionally affected by the drugs, which helps it remain strong and intact. Pharmacokinetic enhancers are prescribed along with antiretrovirals. They help to increase the effectiveness of the antiretrovirals by slowing the breakdown of antivirals. This allows for the antiretrovirals to be prescribed at a lower dosage, which serves to minimize side effects. Considering that HIV does go through an entire life cycle and has a high mutation rate, making it so that there are different strains of the virus in one's body, just one drug will not be effective. In fact, people are generally prescribed multiple of these antiretrovirals in order to interfere with the virus at different stages in its life cycle. This also increases the chance that if one drug isn't quite effective against one particular strain, then another drug will be. Using combination therapy allows a person to minimize the amount of HIV in their system by almost completely stopping the virus from replicating. People are generally prescribed 3 different drugs from at least 2 different classes. In order to determine which drugs to prescribe, genetic resistance testing is done to reveal the types and degrees of mutations that are present in your body. Unfortunately, there is no cure for HIV. This is due to a subset of the virus called provirus. Normally, viruses will reproduce, attack, and spread to infect more cells. This part of the virus, however, does not attack new cells, it simply exists in the body and continues to replicate and divide without agitating an immune response. Essentially, it waits in the body to reactivate when treatment ceases. This is incredibly difficult to target, and until scientists discover a way to isolate and destroy this provirus aspect, there will be no cure for HIV. That concludes my presentation on HIV. I hope you learned a lot. Thanks for listening.